cross. Now, there's some other things we can do following the same sort of logic. So let's take another gene. So let's say gene A is here and gene B is here. Let's say gene D is way down here. Now we already determined this to be 0.5 centimorgans. In this one, let's say this is 34.5 centimorgans. And these obviously aren't to scale, but at least I drew this a little closer than I did these two. But again, not to scale, but the numbers are important. So 34.5 centimorgans between gene B and gene D. And if I asked you to predict the number of offspring, say with a certain genotype, given just this information, you should be able to figure that out. I'm going to show you how to do it now, so you'll be prepared to do it on an exam or a quiz or something like that. So let's say we do set up a genetic cross. Now, such as this, and let's say the question is, how out of 1,000 offspring, so if we have 1,000 offspring, let me sharpen my pencil. (1,000 offspring), how many will have this phenotype? How many phenotype? Let's say big B, big D, little b, little d, big B, little d, little b, big D. So all we really need is this distance, and we should be able to figure this out. How many of the offspring have these phenotypes from this cross given this genetic distance? Well, okay, so what we need to do is determine the number of types of gametes from here and determine which are recombinant and which are non-recombinant. So non-recombinants have the alleles arranged the same as they are in the parents. So B is with a D. So here's a non-recombinant gamete. Here's another one. And now the recombinants have the alleles exchanged. So big B with little d, little b with big D. Okay, so these are all the genetically distinct gametes that can be made by this parent. And this parent over here, it can only make one gametes with one genotype, right? Little b, little d. So when we combine these, we are going to get, let me diagram this with a little more space. Okay, genotypes, BD is going to go up here, little b, little d here, big B, little d here, little b, big D here. Those are the gametes from parent 1. Let me put the genotype of the gamete from parent 2 under each one. And we can see, okay, this corresponds to that, that corresponds to that, that corresponds to that, that corresponds to that. Okay, so the question was, how many of each of these offspring are we going to get in 1,000 offspring? So we were given this distance, so we know, or we can predict, what percent of the gametes were going to be recombinant gametes, right? 34.5%. 34.5% are going to be recombinant gametes. So we would divide this by 2, right? To predict that this is going to be 17.25%. I think that's half of 34.5. And this one is going to be 34.25%. So of all the gametes produced by this individual right here, 34.5% are going to be recombinant. Each one is going to represent half of that, so 17.25%, 17.25%. We can figure out the percentage of these guys too, right? So 34.5% are recombinant, then 65.5% must be non-recombinant. So of the 65.5%, half of them will be this, half of them will be that. So what is 65.5 divided by 2? I think it is 30, it's not 32.5, 32.75, I believe. 32.75%. 32.7, no, that's not right, is it? Yes, okay, yes, I think it is. Okay, so we have the percentage of each gamete produced by this individual, and we've determined that just based on this information up here. We know the distance, genetic distance between gene B and gene D, so we're able to estimate the percentage of each gamete produced by this individual. So back over here again, 1,000 offspring, how many are going to be this phenotype? So we know all of these offspring had to come from a gamete produced by parent 2 here, which is always the same, and a gamete produced by parent 1. So 1,000 gametes from parent 1 made these guys. Of the 1,000, 32.75% were BD. So 32.75% of the 1,000 are going to have this phenotype. Same thing for the next one. 32.75% little b, little d of these gametes were fertilized by this one to get that genotype, to give that phenotype. For this one down here, 17.25% of the 1,000 were this, we're predicting to be this genotype. So those were fertilized by this to give these guys right here, to give off 17.25% of the 1,000 have this phenotype. And same thing for this one right here, 17.25%. So we just need to figure out what percentage of 1,000 is, 32.75% of 1,000. What is the number of offspring from this guy right here? So essentially 0.3275 times 1,000, it's going to be 327.5. 327.5 offspring will be big B, big D. 327.5 will be little b, little d. 172.5 will be big B, little d. 172.5 will be little b, big D. So, okay, great. So that is how we determine genetic distance given, let me see, phenotype information. You saw how we calculated the distance between A and B given numbers of progeny phenotypes. And then we saw how when you were giving a genetic distance, say between gene B and D, we were able to predict the numbers of progeny based on genetic distance information. The numbers of progeny with specific phenotypes based on genetic distance information. So here is a problem. Let's go over 2 quick problems to summarize what we did so far. And so if we take a parent with this genotype right here, and assume these are different genes, different gene A's and gene B's than what we saw in the previous problem. And I give you phenotype information of the offspring from this cross, big A, big B, little a, little b down here, cross to homozygous recessive for both traits over here. And we have progeny, 41, let's say of the offspring are big A, big B for phenotype. 44 are little a, little b. 6 are big A, little b, and 9 are little a, big B. Okay, and so the question is, what is the genetic distance between gene A and gene B? Centimorgans. So what are the centimorgans between gene A and gene B? And so at this point, you can probably pause the video and work on it before I give you the answer. So, okay, what do we need to do? Well, we need to figure out, what are the recombinant gametes made by this individual. And what we would do is, let's see, list them here. Big A, big B. These are the non-recombinants first, and then the recombinants are big A, little b, and little a, big B. Okay, now we can match these up with the phenotypes. So 41 were big A, big B. So 41 big A, big B. 41 of the offspring have the big A, big B phenotype. And we can see that, okay, 41 of the offspring must have come from this gamete right here. 44 were little a, little b. 44 must have come from here. 6 were big A, little b. 6 must have come from gametes with this genotype. 9 must have come from little a, big B. Now, these are the recombinant gametes. And how do we know? Because the arrangement of alleles is different than it is in the parents. For this one and this one, it's different for this one and this one. So these are the recombinants. So we add up those. There are 15 recombinants. There are 85 non-recombinants. So there are 100 total. So the percentage of recombinant gametes is 15 divided by 100, which equals 0.15, and then times 100 to get the percent. We have 15%. So the distance between gene A and gene B is 15 centimorgans. And okay, so that is calculating distance between two genes given the genotypes of the parents and the phenotypes of the offspring. Now, we should be able to do the other type of calculation, which is if we are given the distance between the genes, predict the percentage or the numbers of offspring with each phenotype. So, for example, let's take E and G, genes E and G. Here is the heterozygous parent. And we're going to cross to a homozygous recessive parent. And the distance between E and G, I will tell you, is 22 centimorgans. And so the question I'm going to ask in this case is, how many offspring have small e, big G phenotype? Okay, so easiest way to do this, determine which gametes are made by this individual here, determine the gamete genotypes and the percentages of each. So non-recombinants, and the non-recombinants are up here. So non-recombinant and recombinant down here. Now we're given the map distance, or the distance between E and G, 22 centimorgans. So we know these should make up 22% of the gametes. Each of these should represent 11%. And we're mostly interested in this guy right here, because we wanted to know how many offspring have EG phenotype out of, let's say, out of 1,000. Out of 1,000. So all of the offspring with the little e, big G phenotype must have come from this guy right here. Why? Because this parent only donates little e, little g. So when we combine little e, little g with little e, big G, what do we get for the genotype? We get this genotype, which is, when we translate this to phenotype, small e, big G. Okay. So we know 11% of the gametes have this genotype here. So 11% out of 1,000 is 110. How do we know that? So 0.11 times 1,000 equals 110. So 110 of the offspring should have this phenotype right here. Okay, I hope that wasn't too confusing. I try to get these videos done with one shot, one run through, because I simply do not have enough time to do multiple takes. And I hope if you're confused on anything, please send me an email or come see me during office hours, or make an appointment and we can have office hours outside of the normal scheduled office hours on Tuesday between 2 and 3. Okay, so that's it for that video, and see you in the next video or in lecture.